Okay, so welcome to this next video in which we are discussing P2Y receptors. Okay, right. So we've now discussed the different families of G protein coupled receptors, and we've discussed that the P2Y receptors are all within the rhodopsin family of G protein coupled receptors. What we now want to discuss is when these G protein coupled receptors are activated by their ligands, okay, and we've been through the different ligands that activate the different P2Y receptors. What downstream pathways is it actually going to activate? Okay, so what are they actually going to interact with? Now they're going to interact with heterotrimeric G proteins. Okay, so I now want to discuss the structure of a heterotrimeric G protein with you and the different types of heterotrimeric G proteins. Okay, right. So heterotrimeric then means that we are dealing with a structure which has 3 component subunits. That's what trimeric means. It means it's made of 3 component subunits. Hetero means that the 3 subunits that make up a heterotrimeric G protein are going to be different, okay? G in G protein then means guanine nucleotide binding regulatory. Okay, so G protein is short for guanine nucleotide binding regulatory protein. So this is guanine nucleotide binding. Okay, and then also it's got the word regulatory hidden in there as well. So guanine nucleotide binding regulatory proteins is the full name for G proteins. Okay, right, so let's have a look at the structures of these things then. So if this is the phospholipid bilayer here, basically we'll start off with arguably the most important subunit of heterotrimeric G proteins, which is the alpha subunit. Okay, so I'll draw this here. Right, so basically the alpha subunit can be in 2 states. Okay, it can be in an off state in which it has guanosine diphosphate, GDP bound to it, and it can also be in an on state where it has guanosine triphosphate, GTP bound to it. Okay, so let's initially say that our alpha subunit is going to be in the off state and therefore is going to have guanosine diphosphate bound to it. Okay, so I'll now colour in our alpha subunit of our heterotrimeric G protein in red, and currently it's in the off state, so it has guanosine diphosphate bound to it. Now, another thing that I need to mention about alpha subunits is that they are not integral membrane proteins. They're not sitting within the membrane. However, they are attached to the inner leaflet of the phospholipid bilayer. So the phospholipid bilayer is divided into 2 layers of phospholipids. There is the outer layer of phospholipids which faces out to the extracellular fluid, known as the outer leaflet. Okay, and then there is also the inner layer of phospholipids in the phospholipid bilayer, which faces in towards the cytoplasm of the cell, and this is called the inner leaflet of the phospholipid bilayer. Okay, now basically the alpha subunits of heterotrimeric G proteins are attached to the inner leaflet of the phospholipid bilayer. Now how does this work? Well, basically they have lipid moieties stuck onto the side of them. Okay, so let me tell you exactly what this little line is representing. So, most of the alpha subunits of heterotrimeric G proteins, and there are many different alpha subunits of heterotrimeric G proteins, most of them get palmitic acid molecules attached onto the side of them. Okay, now the more correct name for palmitic acid is hexadecanoic acid. So palmitic acid is the old name for a molecule that would now be called hexadecanoic acid. And although hexadecanoic acid is a bit more of a mouthful, it's a useful name because it tells us exactly what we're dealing with here. It tells us we're dealing with a fully saturated carboxylic acid which has 16 carbons. Okay, so let's draw this structure here. So, here is the carboxylic acid group. Okay, and that counts as one of the carbons in this 16 carbon structure. Then we're going to need 15 more carbons. The final one is going to be in a methyl group. The other 14 in the middle are going to be in methylene groups. Now, because I don't want to have to draw out 14 methylene groups, what I'm going to draw is one methylene group, and I'm then going to bracket it like so, and then subscript it 14 down here. And what that means is take this methylene group and repeat it 14 times. Okay, that will take us up to 15 carbons. We've got 1 here, then 14 here. That ends with the 15th carbon. So the final 16th carbon then will be coming off right at the end in a methyl group here. Okay, so this is the structure then of palmitic acid. Now, some alpha subunits will have palmitic acid molecules stuck onto the side of them. Okay, so they're going to have this carboxylic acid group react with amino acid residue R groups within their polypeptide structure. Okay, and specifically it's cysteine amino acids, but we won't worry about that. Okay, the point is that once you've attached this palmitic acid molecule onto the protein, you have this extremely long hydrophobic tail dangling off the side of the protein, and this is what's known as a lipid moiety. Okay, so a moiety just means something that is present somewhere, basically. It's quite an abstract concept. It just means a structure on the side of something. Okay, so a lipid moiety. Okay, or a structure within something, a part of something, a structure that's part of this alpha subunit. Okay, right. Now, this extremely long hydrophobic tail here will then implant into the hydrophobic core of the phospholipid bilayer, and that will hold this alpha subunit attached to the inner leaflet of the phospholipid bilayer, so it skates about underneath the surface of the cell membrane, basically. Now, most of the alpha subunits do indeed get a palmitic acid molecule attached to them. Some of them get a different type of lipid molecule attached, okay, but it's not too different. Some of them, instead of having palmitic acid attached, instead get myristic acid attached. Now, myristic acid is the old name for a molecule that now will be called tetradecanoic acid. Okay, and again, tetradecanoic acid is a more useful name for this molecule because it tells us exactly what we're dealing with. It tells us that we're dealing with a 14-carbon fully saturated carboxylic acid. So let's draw this now. So here's the carboxylic acid group, and then we want 14 carbons then now. So here's the first one. We then want 12 of them to be in the form of methylene groups. So here's a methylene group, and we want to repeat this 12 times. Okay, and then the final carbon will then be in a methyl group, like so, and that will take us up overall to 14 carbons. 1, 2 on either end, and then 12 in the middle takes us up to 14. So some alpha subunits, instead of having palmitic acid added onto them, instead have a myristic acid molecule added onto them, and therefore this also functions as an extremely long hydrophobic tail, which will then implant into the inner leaflet of the phospholipid bilayer. Now, some alpha subunits even end up with both of them attached onto them, so they get a double whammy, basically. But the overall message is that these alpha subunits are attached to the inner leaflet of the phospholipid bilayer via this lipid moiety that is attached to them. Okay, right. So let's now discuss the other subunits of heterotrimeric G proteins. We discussed the alpha subunit of heterotrimeric G proteins, and we'll discuss the different types of alpha subunit later on. At the moment, we're just discussing the basic structure. Okay, right, so when the alpha subunit is in the off state and has guanosine diphosphate bound to it, the alpha subunit can then also bind to another two subunits. Now, the other two subunits, called the beta and the gamma subunit of the heterotrimeric G protein, these always remain bound to one another. They never come apart under physiological circumstances, okay? And this complex of the beta and the gamma subunit together is often called the beta-gamma complex. Okay, and when the alpha subunit is in the off state, it can associate with a beta-gamma complex. So I'll draw the beta-gamma complex here now. Okay, so this consists of the two subunits, the beta subunit here, and also the gamma subunit here. Okay, so let's colour these in. So we'll have the beta subunit in blue here. Okay, like so. And we'll have the gamma subunit in green. Okay, like so. Now, again, the beta-gamma complex is attached to the inner leaflet of the phospholipid bilayer, and the means by which it's attached to the inner leaflet of the phospholipid bilayer is that it too has a lipid moiety sticking off the side of it, which then implants into the inner leaflet of the phospholipid bilayer, holding the beta-gamma complex at the inner leaflet of the phospholipid bilayer. Now, this is a much more complicated lipid moiety that is attached to the gamma subunit than the lipid moieties that we've seen we can have attached to the alpha subunit. Okay, so we're not going to go into the structure of this lipid moiety. It's what's known as a prenyl group, if you're interested. Okay, but the principle is that you've got this long hydrophobic molecule stuck off the side, and you're basically going to stick this into the inner leaflet of the phospholipid bilayer, and this will hold the beta-gamma complex at the inner leaflet of the phospholipid bilayer. Okay, and when the alpha subunit is in the off state, i.e. it has guanosine diphosphate bound to it, the beta-gamma complex can associate with the alpha subunit to make the full heterotrimeric G protein here. Okay, right. So, what we're now going to do is discuss the different types of alpha subunits, the different types of beta subunits, and the different types of gamma subunits. Okay, and we'll then move on to the G protein cycle, which is how G protein coupled receptors activate the heterotrimeric G proteins. Okay, right, so this is the heavy bit now, when we discuss all the different types of alpha subunits, and all the different types of beta subunits, and all the different types of gamma subunits. And if you're seeing this for the first time, take a deep breath, because there are far more than just S, I, and Q. Okay, right, so we'll start with all the different alpha subunits then. Okay, right, so the first piece of information is that overall there are 16 genes in the human genome which code for alpha subunits of heterotrimeric G proteins. Okay, and when I say the human genome, I don't mean the actual chromosomes within a human cell, because if you look at all the chromosomes in the human cell, you have 2 copies of each of the homologous chromosomes. When I say the human genome, I mean 1 copy of each of the homologous chromosomes. Okay, so if you take 1 copy of each of the chromosomes, you don't have the full diploid number that you have within a nucleus of a normal human cell. You will find 16 genes for alpha subunits that are currently known, of course, and these figures do change over the decades. Okay, this is currently what, as far as I understand, is the correct current view. Okay, right, so there are 16 genes, and this codes for 21 different subunits. Now, that looks an anomaly at first. What? 16 genes coding for 21 different subunits, until you remember that some genes can have multiple splice variants. Okay, so let me just remind you of what that actually means. So we'll do a bit of the central dogma of biology. So let's let these 2 parallel lines here represent a piece of double-stranded DNA. Okay, so we've got one strand of the DNA and the complementary strand here. Okay, and let's say this little region of this double-stranded DNA, which I'm now boxing here, and I think I'll even highlight it in some fun colour. Okay, so we'll have it in orange here. Let's say this represents the gene of some alpha subunit. Okay, and one that specifically has a huge number of different splice variants is the G alpha S subunit, but we'll keep it general at the moment. Okay, so let's say this is the gene for some alpha subunit. Now, one of the strands of the DNA, which let's say is this one that's coloured now in purple, will be called the coding strand. Okay, so let's say this strand that I've coloured in purple, this is going to be the coding strand. Now, what do I mean by that? I mean that this is the strand that the enzyme RNA polymerase 2, okay, often abbreviated to RNA for ribonucleic acid, then we have P for polymerase, and then 2, so RNAP 2, if you like. Okay, so this stands for RNA polymerase. We'll just write P polymerase out in full. Okay, so this is the enzyme which creates RNA from DNA in humans. Okay, this is the strand, this coding strand will be the one that the RNA polymerase 2 enzyme works along in order to create a piece of mRNA. So here it's working along and it's going to create a piece of mRNA. Okay, so this portion in green here, this is a piece of mRNA then. Okay, so this is messenger RNA. Now, the other strand of DNA, which is complementary to the coding strand, which I'll now colour in blue here, this is called the non-coding strand, because this one will not be used by RNA polymerase 2 to make RNA, basically. Okay, so this is the non-coding strand. Now, we therefore have produced this piece of mRNA from the coding strand. So let's bring it out now. Let's say it's here. Okay, this piece of mRNA that has come straight off the production line is not ready to be translated yet. Okay, you cannot just stick this through a ribosome and expect the result to be good. Okay, because there are regions within this that are not supposed to be translated. Okay, so this is what we call the pre-mRNA. Okay, it is the primary transcript that has directly just been produced by copying the DNA, basically. Basically, there are regions within this pre-mRNA that we don't want to actually translate, and there are regions that we do want to translate. So let me box the regions that we actually do want to translate. Okay, so let's say that these portions that I'm boxing now, this portion, and this portion, are the portions that are actually supposed to be read by the ribosome and turned into a sequence of amino acids. Okay, and these are called the exons. Now, the portions in between the exons within the pre-mRNA, this portion here, and so on, these are the portions that are not supposed to be translated, and these are called the introns. So, in a pre-mRNA, you'll have exons and introns, and before we actually translate and produce a piece of protein, we need to cut out the introns and then stitch the exons back together. Okay, and this process of cutting out the introns and then sticking the exons back together is known as splicing. Okay, and you then end up with a shorter piece of mRNA, shown here, that is called the mature mRNA. Now, basically, some genes are capable of producing more than one piece of mature mRNA from their pre-mRNA. Okay, so sometimes there is more than one way of splicing up the pre-mRNA, and therefore you can create different mature mRNAs. Okay, so let me give you a simple to understand example of this. So for instance, let's say that this exon over here is actually optional. Okay, so you don't need to include that exon. Okay, so in some mature mRNAs you will have this exon included, and in others you won't have this exon included. Others will view it like an intron. Okay, and therefore you will have two different mature mRNAs you can produce, one which has this exon and one which doesn't have this exon, and those will code for slightly different proteins, and therefore, because you can create these two different mature mRNAs from the same piece of pre-mRNA, you can create two different proteins from the gene. Okay, and these are known as the different splice variants of the same protein. So basically the principle is that the mature mRNAs that you can get out of a pre-mRNA, it's not necessarily the case that you can only get one mature mRNA out of this piece of pre-mRNA here. Okay, so you can produce multiple different mature mRNAs through different mechanisms of splicing, and I've given you a very easy to understand example there. Okay, but sometimes there are multiple, more than just two pieces of mature mRNA that can be produced, and therefore you can produce different splice variants of that same gene. Okay, so that's how, even though we only have 16 genes, we can produce 21 different alpha subunits. Okay, right, so we'll call it there for this video, and in the next video what we'll do is we'll talk through the 16 different genes for alpha subunits of heterotrimeric G proteins. We won't quite go through the pain of all the different splice variants, mainly because the notation for it isn't quite standardized yet, and there's many different people who use their own notations for them. Okay, so because of the hideous notation for the different splice variants, we won't go there. We'll look at the genes, which are nice and standardized.